Hello everyone, this is Ketish here. In this video, I'm going to go through the AQA Biology Paper 1 Higher Tier from 2019. Question number 8 is on the topic of cell organization or organization. Pancreatic cancer develops when a malignant tumor. What is a malignant tumor? Malignant tumor is a tumor that can actually spread around the body. It's the dangerous kind. A benign tumor doesn't actually do much. It just grows as a lump and it just stays there as a lump. If you remove it, then you got rid of the cancer. So it's far less dangerous than a malignant tumor. The pancreas produces digestive enzymes. What is an enzyme? Well, enzymes are biological catalysts because it's 2 marks, you need to give more detail. Made of amino acids that speed up chemical reactions. Well, a catalyst implies that it speeds up a chemical reaction. This would give you the 2 marks for mentioning it's a biological catalyst and it's made up of amino acids. Those are the 2 marks. Now, you could also say instead of amino acids, you could say it is a protein as well, right? It implies that it's made up of amino acids as well. Carbohydrates is an enzyme produced by the pancreas. Name 2 other organs in the digestive system that produces carbohydrates. Now, carbohydrates break down carbohydrates into glucose. So where else would you actually find? Well, the first one is actually the mouth or in the salivary gland. Name 2 organs. So the mouth is actually not producing the saliva. It's a salivary gland, right? So the marking scheme actually says ignore mouth. Salivary glands, right? And it is also produced in the small intestine. Now, both these organs, the salivary glands and the small intestine, actually produce an enzyme called amylase, which breaks down starch into glucose. Amylase belongs to the group of enzymes called carbohydrates, right? Carbohydrates is not a specific enzyme, right? But amylase is a specific enzyme that breaks down starch to glucose, right? So any enzyme that breaks down any type of carbohydrates into glucose are known as carbohydrates. Now, we commonly come across starch very often, but there are lots of different types of starch or sugars as well, like maltose, sucrose. These are things that we actually consume very often. For example, when you drink 0 sugar Pepsi or Coke, it still tastes sweet. So there is a type of sugar that is not actually doesn't actually increase your blood sugar level, so because it doesn't break down, but it still stays sweet, right? So 8.3, one symptom of pancreatic cancer is weight loss. Okay, explain how pancreatic cancer may cause a person to lose weight. Do not refer to hormones in your answer. So you can't refer to hormones, which means you can't mention enzymes. Okay, if you have pancreatic cancer, now cancer cells reproduce really fast. Which means if they're reproducing really fast, they would be using a lot of energy. So they would consume a whole lot of glucose compared to the other cells around it. That reduces the amount of glucose available for your body to do other things, and one of that is going to be to put on weight or maintain the weight. If there isn't enough sugar in your blood because the cancer cells are using it constantly, your body is going to break down the fat so that it uses that energy, and so which means you're going to lose weight. Now, it also means if you have pancreatic cancer, your pancreas doesn't actually work. If the pancreas doesn't work, the enzymes that is produced by the pancreas, I'm trying not to mention the enzymes here, the enzymes that is produced by the pancreas is going to be less, or there's no enzymes going to be produced by the pancreas. That means the food that needs to be broken down by these enzymes is not going to be broken down easily, which means it's not going to release glucose as well, or it's not just glucose, it's not going to break down the proteins as well. So you can actually talk about specific enzymes because they're talking about hormones. Do not refer to hormones like insulin, but you can talk about enzymes like amylase that I mentioned above. So let's write the answer. There would be less or no enzymes produced by the pancreas. So food is not broken down. You could use the word digested as well, right? Food is not broken down fully. Why am I saying fully? Well, when you eat it, when you eat, your saliva will have amylase. So digestion already starts while you're chewing the food. When it goes into the stomach acid is also going to break down the proteins. It's not completely going to break it down. It breaks it down partially so that it can pass into the small intestine, and the rest of the digestion actually takes place in the small intestine. So it's not fully broken down. So less glucose is released for aerobic respiration, and less amino acids are released from protein digestion. And we need to actually mention about proteins as well, because it's not just glucose which is the source of energy for our body. Glucose is the main one, then it's proteins, then it's fats, right? Fats also release a high amount of energy. So less amino acids are released from protein digestion. This means less glucose passes into the bloodstream, right? So more fat is broken down to release energy. You could say stored fat. Stored fat is broken down to release energy, right? And less muscle protein is made because you don't have enough amino acids as well for growth or repair. That should give you the 4 marks. Enzyme A and enzyme B are involved in controlling cell division in pancreatic cancer cells. Most cancer cells produce both enzymes A and enzyme B. Some people have a gene mutation that stops cancer cells producing enzyme B. Okay, are both involved doesn't mean that they're required. Okay. Figure 9 shows how cell division is controlled in pancreatic cancer cells. You've got pancreatic cancer cells, enzyme A or enzyme B. If it has it, cells divide uncontrollably. If neither of these enzymes are there, cells stop dividing and are destroyed. So you need both enzymes A and B for it to divide. Scientists have developed a drug that inhibits enzyme A. Inhibits means to stop it from working. The drug is given to pancreatic cancer patients who have the gene mutation that stops cancer cells producing enzyme B. So they already have the gene mutation, so they are not producing enzyme B. So they just need something that will stop enzyme A being produced, so that it'll actually stop the cancer cells from dividing and it'll be destroyed. The drug only targets cancer cells. Okay. Explain why the drug can be used to treat pancreatic cancer in patients with the gene mutation. Use information from figure 9. Now, figure 9 says enzyme A or B is required. So, or B, not and. That's key. Right? Cancer cells repeat the word. Repeating it again. Cancer cells cannot divide and are destroyed cancer cells cannot divide and are destroyed if or when both enzymes A and B are inhibited. Right? So there going back to the question before I continue, explain why the drug can be used to treat pancreatic cancer in patients with the gene mutation. They already don't have enzyme, or their body stops them producing enzyme B, and the drug will stop the enzyme A. So cancer cells cannot divide and are destroyed when both enzymes A and B are inhibited. So there is no chance of the tumor growing and is most likely to get destroyed. Right? Looks like I'm actually repeating it, but you do need that clarification in your answer for the third mark. Explain why the drug could not be used to treat pancreatic cancer in a patient that produces both enzyme A and B. Right? Drug only inhibits A, not B. I'm just being too lazy to write the word enzyme, but still me implies I'm talking about enzyme A and B. Drug only inhibits A, not B, since, or can I say since, right? Let's reword that. Drug only inhibits A, not B. So cells would still divide uncontrollably when enzyme B is still produced by the body. So it will not kill the cells as well. The drug was trialed before it was licensed for use. To improve validity of the results in the trial, some patients were given a placebo. Now, placebo is a dummy tablet, which is basically a sugar tablet. Maybe not sugar, because if the patient actually tastes the sugar, then they might actually know it is a sugar tablet. It is just basically chalk. It doesn't actually, maybe just very similar taste to the real tablet, because most chemicals that are found in tablets, the medication, don't actually have that much of a taste. Right? A double-blind trial was used. What does a double-blind trial actually mean? Now, you have patients who get the tablets or the new drugs. The patients don't know anything about what type of drug that they actually get. Well, who's going to give the patients the drugs? The doctors. Now, the doctors don't know as well what type of drugs, or whether is it a placebo or the real drug that is actually being given to the patients. Why? Because the doctors might actually give patients, certain patients the real one, and certain patients the fake drug. So there would be bias. So who's giving the drug? It's an independent person or a company which actually provides the drugs straight to the patients in most cases. So the doctors are not actually involved, but the doctors are only involved in monitoring the patient's health and looking at the results, not the drug administration itself. So the doctors would actually gather results and pass it to the companies, to the same company or to a different company, and the results are put together to form the final report. This is what a double-blind trial actually means. Give reasons why a placebo and a double-blind trial are actually used. Like a placebo is used to avoid bias, results, actually let me rephrase that. Placebo is used sorry, let me get rid of that. A placebo is used as a control for comparison a double-blind trial is used to avoid biased results. One stage in a drug trial is to test the drug on healthy volunteers. What is the next stage in the drug trial? Testing on all patients with the disease? Testing on human tissue? Well, one stage in a drug trial is to test on healthy volunteers. What is the next stage in the drug trial? Once you have tested it on healthy volunteers, there's no point in actually testing it on human tissue. You would actually test it on human tissue first. Then you would actually test it on live animals as well. Right? Tissues are actually relatively cheap rather than animals, and ethically it's better. So you would actually do that first. Treat it with on live animals, right? To find out how much would actually kill the animals or if it actually has any side effects. Then you would test it on healthy volunteers to see if there's any side effects. Then you would test it on volunteers with the disease. Finally, you test it on all patients. So the next stage is testing on volunteers with the disease. A monoclonal antibody can be produced to treat pancreatic cancer. Explain how the monoclonal antibody works to treat pancreatic cancer. A monoclonal, mono means one, clonal means identical. Antibody, what do antibodies do? They attach themselves to specific areas of a cell or the targeted. So you could produce the monoclonal antibody and inject it. The antibody will move around the body, but they will only attach themselves to the pancreatic cancer cell. So antibodies are basically these Y-shaped chemicals. And let's say you have a cancer cell and it has this specific shapes on its surface area, on its cell membrane, or these are protein molecules. The antibodies I'll write in a different color, will attach themselves onto these. So they will be targeting just the cancer cells, because these spiky things are only present in, let's say, in the cancer cell. That way you can target it. Well, how is that going to actually help? What if I attach a radioactive isotope or compound to this antibody? That way, when the antibody actually attaches itself to the cancer cell, that cancer cell is going to be surrounded by radioactive isotope, which is going to release radiation, could be alpha, beta, or gamma. That radiation can actually kill off the cancer cell, right? So the monoclonal antibody is attached to radioactive substances, or you could actually attach toxins as well, or toxins, or even drugs, right? Well, certain drugs are toxins, right? The monoclonal antibody would only attach to the target cancer cells. So the radioactive substance or drug can stop the cancer cells from dividing, or you could say it could actually kill them as well. That brings us to the end of this question. Question number 8 is actually out of 19 marks. That's nearly 20% of the paper. That's a big question. And that's a lot of marks as well. But it's a very good question to answer as practice. That brings us to the end of this video and to the end of this paper as well. You should be able to find the other papers in my YouTube channel. If you did find this video useful, consider subscribing to the YouTube channel. I've got videos of me going through biology and physics, and I'm trying to put the chemistry papers as well as part of to help you for revision. Thanks for watching, and I will see you in the next video.